we're going to start working with electrodes. What I'm going to be focusing on are two things. Invasive electrodes that can go inside the body, and the other one is surface electrodes. We need to understand an electrode-electrolyte interface. See guys, this pen, that's AgCl, which means this is silver chloride, right? Now, inside the gel here, in this gel material, we should have ACl ions alone and chloride ions alone. So whatever gel you're making it from, it has to have in it ions from the same material of this metal. How come we are putting it on the body and it's sensing electrical signal? There should be something happening inside the gel that allow us to measure. In order for it to work, the liquid or the gel that's here should contain ions made of the same material of this pen. The electrode, this is the electrode, right? And then we have something called the electrolyte. We need to understand this electrode-electrolyte interface in order to be able to assess how we are actually using it to measure the biopotentials. So let's say we have AgCl or platinum or polymer coat. There should be a flow of current between the electrode and the electrolyte. The black rectangle is your electrode inside the body. What is the cause of electrical current? They're the ions, correct? In the electrode, in this material here, it's metal. The source of current is the electron, not the ions. Let me repeat that again. Your current flow inside the body depends on the ions, positive or negative. In the electrode itself, it's connected to a wire, right? You will have a wire connected to it, and the wire is made of metals, so there's a lot of electrons moving. So we have a current is passing between the two, but one side electrons and the other side ions. The electrode is made of conjunction of salt. The movement of ions inside the body create a current. The movement of electrons in your electrode and the wires makes the current. Both of them should be the same, but one is made of electrons and the other one made of ions. So let's say I have something called electrode-electrolyte interface. I have the electrolyte, which is this material, the gel. I have the skin, and then I have the body. I have a lead wire, that's my electrode, which is this metal thing, and then I have the electrolyte gel. This gel is the one that's here. Whatever this metal is made of, the same ions should be in the gel. And then you have the skin, and then you have the body. I would like to study the interface between the electrode and the skin, as a whole, so the entire thing. But I can break it down into two sub interfaces: electrode-electrolyte and electrolyte-skin interface. So I can study this interaction here. See this layer that I'm doing this red line on it? I need to study this interface, and I need to study this interface. So I'm looking electrode, electrolyte, and electrolyte skin. Do we good? We have two things to look at here. So what is happening? First, the electrolyte skin interface can be visualized and understood as the skin acts as a diaphragm or insulator between two solutions. The electrolyte, this gel material, is a solution, right? It has ions. Those ions can move. And also I have the skin, beneath the skin, I have my body fluids, and they have ions. The skin is a barrier between two solutions. Do we agree? You have the body fluids, and then you have the electrolyte. The skin, which is the yellow here, is an insulator. So each contains cations and anions. What do we mean by that? Which one is positive? The cations are the positive ions, and the anions are the negative ions. We have two liquids, and each of them has ions. Let's say I have positive ions aligned on this surface. What should be happening on the other side? You will have negative ions, right? Aligning, correct? Say the different charges, they attract each other. Now, if you have two negative on the top, and you have an insulator, and you have positive below it, it's just like a capacitor, correct? If I had a voltmeter between this point and that point, there should be a voltage that is developing. This gives rise to a potential of non-physiological origin called the contact potential. If I have this electrode, I place it on my skin. I'm not doing anything with my hand. Because of the ions in this electrolyte and the ions in my body fluids, they will align across the skin. And then I have this potential across the 2 points, outside and inside. Does it make sense? If there's no excitation, this is called the contact potential. From its name, because of contacting just the electrode to the skin, there is a voltage actually developing there. The reason why this is happening is because we have ions in this electrolyte, this material, and I have ions in the body. And there's a skin that's preventing them from contacting. Then they will align. It's very small, very subtle, but it does exist. Now, if you have here negative charges, the electrolyte is filled with ions, correct? The AgCl ions. There will, if you have a line of negative charges, there will be another line of positive charges on top of it. And on top of this positive line, there will be another negative line. So what happened is that it looks like I have ions that they're arranging themselves as layers. Do you see that? See your electrolyte, see this material inside it? All the ions in it, they will be line by line. So positive, negative, positive, negative, positive, negative. So it will arrange them, they will arrange themselves this way. As long as there is no activity under the skin, that arrangement will continue. The moment things happen here, inside the body, this is going to disrupt the ions on the surface, which lead to disruption of the ions on the top of it, then disrupting the current flow in the metal. So electrode-skin interface, we talked about that, and then we have the voltage. So the type of potential at the electrolyte-skin interface. The blue is the electrolyte, correct? The yellow is the skin, and then the red is the body, and then we have the electrode, which is AgCl. Contact potential. What is the contact potential? It is the potential that develops between outside the skin and inside the skin without any biological activity taking place. Biological signal of interest, when any disruption happen inside the body, which is an action potential, will result in changes in that alignment of the ions, and that will result in changes in the current. And that's how we're measuring from these electrodes. They're very small, very, very small changes, but we can detect those. So it's either contact potential, or we have something that is happening in the body that will disrupt this alignment, and that can be sensed on the other side. What happened in the electrode-electrolyte interface? Remember the blue is the electrolyte, right? So it's just this one. The ions in the electrolyte will align, and when they come to the electrode itself here, the electrodes are mostly metallic. At the metal-electrolyte interface, the metal discharges ions into the electrolyte, and the ions in the electrolyte tend to combine with them. So see this interface here? That's very important. See this layer, this thin layer of the electrolyte, of the electrode, the metal. We know that if the electrolyte is made of the same ions of the electrode, we will have some ions released from this metal into the electrolyte. So if it's AgCl, right, at the interface, some of Ag from this metal will go inside here, and some Cl. And they will start combining with the other ions in this electrolyte. Now that change, if you're losing positive, right, it looks like there is a current passing from this metal to the electrolyte, and that is going to make an electron release to go inside the wire to the device. there's one condition. This electrolyte should always have the same ions from that metal. If it's AgCl, it has to have Ag and Cl. And that only happen only in the interface, at the interface. So this, see this metal, this electrode here? If you go on the other side, it is going to be losing some of its ions into this gel. So that will be losing ions into the gel. The moment it loses the ions in the gel, based on that disruption, will create a current of electrons that's passing up, and that's how we measure into the device. This develops a charge layer or gradient at the interface called the electric double layer. So that's another term. Contact potential between the electrolyte and the inside the body. The electric double layer happen between the electrode and the electrolyte and the gel. You need very, very small number of ions to move in order to be able to detect that changes in voltage. And that's how we measure the EEG and the EMG and the ECG using this surface electrode. It's all about the alignment of ions inside the electrolyte gel. This potential is called half cell potential, which is the electrode potential. You remember we have electrode, electrolyte, and electrolyte skin. So we have two things, two potentials. Half of them is the electrode electrolyte potential, which is between this layer here and the closest layer to it. So that's called the half cell potential. Between the electrode and the skin is the other half. All electrode potentials are measured in with respect to a reference electrode, usually the hydrogen absorbed in platinum black. So there is a way for us to calculate how much that half cell potential with reference to a standard hydrogen and platinum. So this is AgCl, right? That this electrode is AgCl. We have another electrode that we can make from hydrogen and platinum, and that's our reference that we measure, against. Okay, so let's summarize. You have an electrode, correct? Placed in hydrogen in a gel. Make sense? That electrode is AgCl. That gel has ions of Ag and Cl. We good? That's surface electrode. Now, if you place this on the skin, you'll have two things. You have electrolyte interface, and then electrolyte skin interface. We good with these two? Right? You will have a potential between the electrode and its electrolyte called the half cell potential, and then you have a potential between the electrolyte, the gel, and the skin, and then whatever happened inside the body will be sensed through the different layers of ions. If something changes in the body, that will disrupt those layers, and those layers will then change the flow of electrode into the electrode. So that's how it works. So we need this metal, we need the gel, and we need the skin to be able to measure. The entire action happen in the electrolyte. See, in this blue, where we have AgCl, this gel, that is the most important one, right? How the ions are arranged and how are they disrupted will measure for us the electrical activity under the skin. Now, you can look at some equations between the electrode electrolyte. Again, the electrode electrolyte is here, just gel, that metal. Now, you can have C, which is AgCl, right, as a compound, and if you break it down into the positive and negative components, you can have free electrons. So if an electron has same material as cation, electrode has the same material as the cation, then this material gets oxidized. Do you know what oxidation means? It's losing electrons, right? And yes, so get oxidized and enters the electrolyte as a cation, as an electron, a negative charge, and electrons remains in the electrode and flow in the external circles. So let's think about this way. You have your electrode, right? And this is the gel. So let's try to represent this. See, the blue is the gel, correct? The red is the metal. So what are we saying? In an electrode has same material as the cations. The cations are the positive, right? Right. So if I have Ag here, then this material gets oxidized and enters the electrolyte. So whatever on the surface here, we will get Ag positive inside the liquid from the electrode, and then I have a negative charge left inside the metal. That negative is an electron, and that's going to go up into the wires because there's no need for it. So just AgCl, right? So if you took the Ag into the liquid and you have a negative charge chloride here, we need to keep it balanced. An electron has to be released and move up into the wire. And that's how we create the signal into this electrode. Now, if an ion can be oxidized at the electrode from a neutral atom, or 1 or 2 electrons are given to the electrode itself. So I don't want you to get into the chemistry of it, but here's what I'm saying. If the ions of the electrolyte are arranged in layers, that's good. If we have action potential that will disrupt this arrangement, that disruption will result in electrons passing into the wires. That's it. That's the whole idea that we would like to keep in mind. Right, and then if you want to use the oxidation loses loss of electrons, and reduction is gaining of electrons. What is happening is always we are having oxidation into the metal itself, and that's why we are getting electrons that passes through the wires. Because ions do not pass into electrical wires, correct? It's electrons, but in the body it's ions that generate the currents. Right, we good? The most important thing, are they arranged into this hydrogel? The disruption of the arrangement will result in changes into the electrode-electrolyte interface, and that's the current that we're going to measure. It's going to result in electrons moving, and that's how we measure it. Okay, half-cell potential is a characteristic potential difference established by the electrode and its surrounding electrolyte. Remember, the half-cell potential is between the electrode, this metal, and the gel, and depends on the metal, concentration of ions in solution, and temperature. So these things could be expensive or cheap depending on what material we're using and how many ions in the gel. That's it. That's how we decide those things. And the temperature as well. Half-cell potential cannot be measured without a second electrode, and that's why we need to have 2 electrodes in order to measure a voltage. The half-cell potential of the standard hydrogen electrode has been arbitrarily set to 0. Other half-cell potential are expressed as potential difference with that electrode. So the hydrogen is our reference. Reason for half-cell potential: charge separation between the skin and the body, and then the arrangement of the ions, and any disruption because of the action potential will disrupt those layers. That's the most important thing. Now, I don't want you to get into the chemical reaction of how did we measure the half-cell potential, because that's chemistry, and I don't want to go there. But I want to show you this. I want to show you that different materials will result in different half-cell potentials, right? We would like for the half-cell potential, in order to be able to use it, we want it to be positive actually. So that's why we're using AgCl here. we need to be positive and small. because we would like for it to sense the small changes in the voltages. Very stable, used in ECG. That's why we're using it. Ag is small, half cell potential, and positive. I don't use the other one because that's not what we want it to be. Make sense? We're good with that? All right. I want to stop here because I would like to show you a couple of examples before we go into the polarization of these electrodes. But to feel better, to feel good, I just want to make sure that we are on the same page. We have electrode-electrolyte interface, and we have electrolyte-skin interface. We good? The arrangement of ions inside the body and in the electrolyte determine how the current is passing into this electrode. Very small values, but we can sense them. And we can magnify them, and then you can see this EEG on the screen because of that. It's a very smart idea, actually, to be using these things. And it's cheap as well. Very, very cheap to build that thing. We good? Okay, let me stop here. And on Friday, we will go over the exam material and solve problems so that you'll be ready on Monday for the exam. Make sense? Okay, let me stop this.